Are there any questions about the groups of Protista that we talked about at the end of last week? Or anything else we've discussed recently? Okay, well, if not, we're going to go ahead to screen sharing and continue looking at groups of Protista. Keep in mind a couple of the things that I said last week. One is, as we look at micrographs, photographs taken through the microscope of the various groups, you will notice that a lot of the groups look very similar, and this helps to explain why it was very difficult to classify Protista when really all we could do was look at them through a microscope. They don't have enough differences in their appearance when we look at them through a microscope for that to be very useful for us. The other thing to keep in mind is, as I said, we would be looking at these slides in lab. So it's not quite the same as having you adjust the microscope or anything, but it's as close as we can come under the current circumstances. So the next group we're going to look at is the apicomplexans. And the apicomplexans are a group that are what are called obligate endocellular parasites. Obligate means they have to survive this way. And they're parasites, but they're endocellular, which means they live inside cells. And their name is really a combination of two words. The apex, or the apical end of something, is the very tip. And they have a complex at this end. And the complex is really just some unique cell organelles. Now, since these are parasites, they cause some fairly serious diseases. In a good year, they only kill about 500,000 people around the world. Many years, they kill closer to 2 million. We're going to focus on a genus called Plasmodium. Plasmodium by itself kills more than 400,000 people. Plasmodium is the genus of Protista that cause the disease that we call malaria. There are several types of Plasmodium. Some of them cause more severe types of malaria than others. And like a lot of parasites, we've already seen some examples, but Plasmodium has a very complex life cycle. And this is something we often see with parasites. They have a life cycle involving more than one host, more than one organism that they live in. And they have a life cycle that involves several different stages that we could look at under a microscope or sometimes even without a microscope and distinguish. In the case of Plasmodium, Plasmodium's second host is a mosquito of the genus Anopheles. And it's only females that Plasmodium lives in. Female mosquitoes are the only mosquitoes that bite you, by the way. Male mosquitoes eat nectar from flowers. Female mosquitoes suck blood because since female mosquitoes lay the eggs, they need extra protein to build the eggs. So they get that extra protein by biting you and sucking some blood. So let's look at the life cycle of the malaria parasite. And we're going to start up here, or I should say down here. Let's go down to the bottom. We're going to start down here with a stage of the life cycle called sporozoites. Sporozoites literally means spore animals. Now, sporozoites live in the salivary glands of a mosquito. And when this mosquito bites you, what they do is before they bite you, they inject some saliva into your skin. You may not realize this, but if we look at your skin, the nearest blood vessels to the surface are about 25 cell layers deep. There are no blood vessels closer than 25 cell layers to the surface. So for a mosquito to get to your blood, they've got to go through 25 layers of cells. So the saliva has some pretty nasty stuff. It has some acids, it has some materials that basically help it to dissolve these first 25 layers of cells. And when it dissolves its way down there, the mosquito's mouth actually looks kind of like a needle. So it pokes you, it gets as close to the blood as it can, releases its saliva, the saliva dissolves cells the rest of the way down, and then the mosquito pokes this needle-like mouth part into your blood, and it sucks up blood cells. But the saliva that it injects into you contains sporozoites. Now, these sporozoites can't really swim, they just kind of drift on the blood. And when they get to the liver, they enter a liver cell, something called a hepatocyte. Hepat means liver. So these sporozoites enter a liver cell and they start to reproduce. And when they reproduce, they form a stage called merozoites. We're going to have a lot of stages here, and they're all going to end in zoide, unfortunately. Well, all except one or two. Now, these merozoites increase inside the hepatocyte, and eventually these merozoites break out of the hepatocyte. They go back into your blood, and they circulate in the blood. And again, they can't really move on their own. They just drift with the blood. They move because of the pumping action of the heart. And these merozoites can enter a human blood cell. When they enter a blood cell, one of two things happens. They can either reproduce inside the blood cell, and they reproduce just by mitosis. So notice that means all the merozoites coming out of this blood cell are genetically identical. Eventually, if they're reproducing inside the blood cell, there are so many of them that the blood cell just bursts. It ruptures and releases merozoites out into the blood again. When the red blood cells burst, for a person who has malaria, this is when they have their bout of fever. This is when they show the symptoms that we tend to associate with malaria. Now notice that this can potentially cause problems also because remember, red blood cells are carrying oxygen from your lungs to other parts of your body. So as merozoites destroy red blood cells, you may find it hard to deliver oxygen to parts of your body. So you may have a form of anemia very temporarily. So that's represented by this little circle off to the side of the main circle. This is the merozoites reproducing inside a blood cell, making more merozoites. However, the merozoites can also differentiate into gametocytes. Now, gametocytes, you know that gametes are the eggs and sperm. So there are male and female gametocytes. We don't usually call them eggs and sperm. They used to call them that just kind of because we thought of everything in human terms, but we've realized in recent years that they're not really the equivalent of eggs and sperm. So gametocytes are released from the red blood cells and they're circulating in the blood. So when this mosquito bites you, it injects sporozoites, but if another mosquito bites you, it'll inject some sporozoites, but it also picks up gametocytes. So over here on the right-hand side of the big circle, this part that's in kind of the tan color, these are the stages of the life cycle that occur inside the mosquito. Over here, the red is the parts that occur inside you, inside a human. So the gametocytes develop into gametes. Two gametes fertilize each other and form an ookinete. Now, all this is happening inside the mosquito's digestive system. The ookinete burrows through the walls, through the cells that make up the mosquito's digestive system. They get into the mosquito blood and they travel to the salivary glands. And while they're traveling to the salivary glands and while they're in the salivary glands, this ookinete develops into an oocyst, and the oocyst makes sporozoites. So we're back where we were. But notice we have sporozoites, we have merozoites, we might have more merozoites, we have gametocytes, we have gametes, we have this ookinete, we have the oocyst, we have sporozoites again. So we have a lot of stages in the life cycle. Now notice that, like most parasites, and we'll talk about some more as we go through the semester, when Plasmodium is in the mosquito, it doesn't really hurt the mosquito. And this is what we see for many parasites. They have one host where they cause problems, and they have another host that they don't really cause too much damage to. The mosquitoes that are carrying these oocysts or sporozoites, whatever stage they happen to be in, as far as we can tell, it doesn't hurt the mosquito at all. So the mosquito doesn't get any disease that's the equivalent of malaria. Now, there are many other parasites in this group. And when we look at these parasites, in general, they all can only move the same way that Plasmodium moves. They have to get inside a host, and they have to let the host's body fluids carry them around the body. The organisms that are in this group are not capable of independent movement. They don't have cilia or flagella. They don't put out pseudopodia. So the only way they can move is if the host's blood carries them or some other type of body fluid. They don't all live in blood, but most of them do. So when we look at this, you might remember that some of the drugs that President Trump proposed for using to treat COVID-19 originally were drugs that have been used to treat malaria in the past. Unfortunately, most of those drugs have a pretty minimal effect. It looks like some of them may lessen the severity of your symptoms, but none of them appear to do anything to stop you from actually getting COVID-19. And unfortunately, he was proposing it as being like a vaccine, that you wouldn't get it if you were taking these drugs. So when we look at these organisms, notice they were originally classified as protozoa, protista that resemble animals, even though they couldn't move because of the fact that they were floating in the blood, I suppose. So when we look at these organisms, keep in mind that, like a lot of other protista, these are good examples of something that its biology is very different from human biology. So this is another reason why trying to fit everything into the kind of the processes that we see in human biology doesn't really work. These life cycle stages, the merozoites, the sporozoites, the ookinete, these are not similar to a baby and a toddler and a child and an adolescent. These are very different things that if you looked at under a microscope, you wouldn't necessarily know they came from the same species even. Now, the next group we're going to look at are the ciliates. And as you can tell by the name, the ciliates have cilia that allow them to move, at least during part of their life cycle. Some of the ciliates lose their cilia and only have it for certain periods of their life cycle. So if you look at the notes, you should be able to see the cilia. I'm not sure there's enough contrast in this photo for you to see the cilia on my screen, but the cilia are a bunch of hair-like structures. In the case of Paramecium, they really cover the entire cell because keep in mind, this is a single-celled organism. These cilia have that same 9+2 structure of microtubules. And remember, microtubules are made from tubulin proteins. So this is the same structure that we saw in flagella. And this is one of those things in biology that we don't really have a sharp line that separates cilia from flagella. cilia are generally shorter than flagella. Cells that have cilia usually have lots of cilia. Paramecium typically has hundreds, sometimes thousands. Cells with flagella, excuse me, usually have maybe one, sometimes 4 or 5, but they don't have hundreds of flagella. But if we took a cilia or a flagella, remember this is kind of a long stringy thing. If we took it and sliced through it, we would see the same 9+2 structure. You wouldn't be able to look at a cross section, a slice through a cilium or a flagellum and tell which one you were looking at. Now, keep in mind that ciliates, and paramecium is a good example of a ciliate, have some of the largest cells in the protists. And their cells are so large, in fact, they're larger than a lot of human cells. And this is going to explain something that we'll see in a moment. Because they're so big, ciliates have a very large polyploid macronucleus. Macro just means big. So it's polyploid. So what does that mean? Well, remember when we talked about sympatric speciation, we talked about polyploidy being one of the ways that that could happen. And a polyploid nucleus isn't haploid, it isn't diploid, it's triploid or tetraploid. It's got multiple copies of the chromosomes. And the reason it has multiple copies, the reason that this cell is polyploid is because it's so big, because this functions in what are usually called non-reproductive activities. But if you go back to what we talked about in Biology 105, non-reproductive activities means protein synthesis. So, you know, if you're a really big cell, the advantage of having multiple copies of your chromosomes is you can make more proteins simultaneously. If you only have one copy of a chromosome, you can basically only make one piece of messenger RNA from that at a time. If you have multiple copies, if you have 6 copies, you could maybe make 6 copies of messenger RNA from the same gene at the same time. So it's easier to make the large number of proteins. Now, the other thing that's unusual is these organisms have one or sometimes more than one small micronucleus. And the micronucleus, micronuclei, I should say, since there's more than one, are diploid. So notice that the 2 nuclei don't have the same number of chromosomes. If one of them has 10 chromosomes, the other might have 20 or 30 or 40. It's polyploid. The micronuclei function in reproduction and horizontal gene transfer. So remember, when we talked about bacteria, we discussed horizontal gene transfer, changing the DNA of cells without reproducing. So reproduction in paramecium is usually by binary fission. And it's very similar to what we saw in bacteria. But the key word is similar. It's not exactly the same for some reasons we'll see in just a moment. So here's a paramecium getting ready to divide by binary fission. And what we see is that the macronucleus divides amitotically. Now, when we say that the macronucleus divides amitotically, A at the beginning of a word means not or without, like asexual. So remember, this macronucleus, this large oval thing here, this is polyploid. It has lots of copies of the chromosomes. So it divides, and it divides basically just by stretching. The chromosomes that are in the top half are going to end up in a cell on the upper portion. The cells that are in the bottom half of the macronucleus are going to end up in the lower cell. So notice there's no attempt to divide up the chromosomes evenly. Since the macronucleus is polyploid, usually they get divided up equally, but not always. Every once in a while, this process creates genetically different cells simply because the chromosomes that are in the upper part of the macronucleus end up in the upper cell. The ones in the lower part of the macronucleus end up in the lower part. Now, then, if we look at the micronucleus divides by mitosis. And so this is showing spindle fibers here, and then there's a little circle at the top and bottom. Those are the two micronuclei that have been created by mitosis. And then we have the cell pinching inward, and the cell pinches in, and it forms new contractile vacuoles. It forms a new oral groove, this kind of yellow part, which is where it funnels the food into. It replicates organelles in the cytoplasm. So notice that this fourth picture over here is similar to what we talk about when we're looking at, say, human cells, when we talk about cytokinesis. Now, eventually, we have two cells, and the two cells separate, and they go their own way. If we look at a microscope slide of paramecia in fission that's been stained, here we have a plain paramecium over here in the lower right. Notice you can see the macronucleus, this large dark purple oval. The greenish are the cilia. All the lighter purple around the macronucleus is the cytoplasm and the organelles. If we look here in the upper left, you can see a cell that's just starting to go through binary fission. How can you tell it's just starting? Well, if you look at this indentation in the middle, this indentation hasn't gotten very far. If you look in the upper right, you can see a cell that's a little bit farther in binary fission because that indentation is going in deeper. And if you look down here in the lower left, you can see a cell that's almost done with binary fission. Maybe hard to see on my screen, but if you look on a copy of the notes, if you download it or look online, there's just a very thin connection between these two cells. So the next stage is they'll separate completely, go their own ways, and then eventually they may divide by binary fission again. Now, horizontal gene transfer in paramecium occurs by a process called conjugation. And it originally got its name because it looks somewhat similar to conjugation in bacteria cells. Remember, in bacteria cells, we had two bacteria cells kind of line up next to each other. They made a cytoplasmic bridge from one cell to the other. And if we look at paramecium, we see something similar. We have two paramecia that line up next to each other. They kind of align along their oral grooves, their feeding structure. So they don't feed while they're going through conjugation, but the cilia still move even though they're in conjugation. You can see paramecium swimming through the water just fine. Notice over here, we have some long algae next to the paramecium, but we can see two pairs of paramecium in conjugation here. So let's look at this process, and I'm going to magnify this a little bit. So we have our nuclei shown in purple, and the macronucleus is up on the top. The micronuclei are down on the bottom. Notice they're not generally separated that widely when we look at an actual paramecium. That's more for convenience. If we look at what happens, remember we said there's more than one micronucleus in a cell. All of the micronuclei, except one of them, deteriorate. they fall apart. The materials in them are absorbed into the cytoplasm and can be used for building other materials. Once that happens, the one micronucleus that's left divides by meiosis. So in our next diagram, we have 4 little micronuclei. Now, 3 of these micronuclei disintegrate. So I know, why bother to go through meiosis? Well, this micronucleus over here in the first diagram is diploid. After meiosis, this micronucleus in the second diagram is haploid. So all of the micronuclei except one of these little orange ones and one of these little purple ones disintegrate. Once that happens, the remaining micronucleus goes through mitosis. The remaining purple micronucleus goes through mitosis. So now we have 2 little genetically identical haploid orange micronuclei. We have 2 genetically identical purple micronuclei over here. Now what happens is, so this is where our gene transfer comes in. One orange micronucleus goes from the cell on the left to the cell on the right. One purple micronucleus moves from the cell on the right to the cell on the left. So we end up with a paramecium cell that has one orange micronucleus, one purple micronucleus. Same thing over here, notice. Both of these micronuclei are haploid. So those micronuclei fuse. So now we have a diploid micronucleus, which remember was what we started out with way back over here. And once this micronucleus is diploid, now it divides and it makes a macronucleus and it makes more micronuclei. Different types of paramecium, different species have different numbers. So it makes whatever the appropriate number is for this particular species. And now we have 2 cells at the end, 2 paramecium. Notice that these 2 cells are genetically identical. They both had an identical orange micronucleus and an identical purple micronucleus. But they're identical, but notice they're different than the cells that we had at the beginning, because this purple micronucleus divided by meiosis, this orange micronucleus divided by meiosis. So at the end, we have 2 new paramecia. They're genetically identical to each other, but they're both different than the cells that started conjugation. Now, we didn't make a new paramecium. We started with 2, we ended with 2. So that's why this is not reproduction. But we changed the DNA of the cells. So that's why this is horizontal gene transfer. Now, the next main group we have is the Rhizaria. Remember, what we're talking about now is the SAR clade, the Stramenophilus, the Alveolata, and the R is the Rhizaria. And the Rhizaria look very different from each other. But like a lot of the Protista, the reason they're in this group is because of genetic analysis. Genetic analysis reveals that they're a monophyletic group. Now we know mono means 1, so that basically means there's 1 common ancestor for all the Rhizaria. One of the things that they have in common, and that we can see very well in the foraminiferans, is most of the organisms in this group have thread-like pseudopodia. Now remember, we saw pseudopodia on amoeba the other day. Thread-like pseudopodia literally are like threads. They're very narrow structures. In foraminiferans, they use these to capture their food. Now notice, if you would go swimming in the ocean or in a lake, there are some forams that are freshwater, most are marine. You wouldn't see most of the foraminiferans in the water because most of them are benthic. And what benthic means is they live on the bottom of the body of water. And a lot of forams are actually buried under a couple of centimeters of mud or sand or whatever's on the bottom of the ocean bed where you happen to be. So you actually have to take kind of a soil sample to actually see them. Almost all species do something unusual. They make an internal shell. And this internal shell is called a test. Now, what's unusual about it is if you look at most animals that have a shell, the shell is outside of the cells. Now, foraminiferans are single-celled, but their shell, their test, is actually located inside the plasma membrane. So it's in the cytoplasm. A lot of species have tests or shells that have multiple chambers. And the different cell organelles are kind of divided up and located inside these chambers. So here's some fossils of foraminiferans shells. And if you look at this large one that's kind of in the center, you may be able to see these little kind of curly grooves in it. Kind of looks like a snail shell, but these are actually different chambers in the test. And since the test is inside the plasma membrane, these chambers divide up the cytoplasm. Now, the tests themselves can be made of calcite, which is a type of calcium carbonate. They can be made of protein, or in some of these foraminiferans that live at the bottom of lakes and oceans, they're actually made from small particles that are found in the sediment where they live. Another group of rhizarians is the cercozoans. Most of the cercozoans are either amoeboid cells or flagellates. And again, they tend to feed by their pseudopodia. And they're so small, again, they're single-celled. They're predators, but what they prey on, what they eat is bacteria cells. And they don't even really eat big bacteria cells. They tend to eat small bacteria cells. And this group looks very different from one another. There are several subgroups. Of course, people don't agree on exactly how many, but if we do a molecular analysis of their ribosomal RNA and some of the proteins like actin that are found in the cells, we can tell them apart. So here's an example of a cercozoan. Again, I don't think you'll be able to see it on my screen, but if you download the notes or look at them online, you might be able to. There's a single flagellum out here behind it. So these organisms are very similar to some of the other ones we've seen. And again, this points out the difficulties in classifying Protista before we could actually analyze proteins for the amino acid sequence and before we could analyze DNA for the base sequence, the ACGT order. Now, the next group of organisms we're going to look at is the organisms that are classified with plants. Later in the semester, we'll look at the kingdom Plantae, the plants. That'll be the last kingdom that we'll look at simply because hopefully the weather will be nice by then and you can actually get out and see a few plants and observe some of the things that we're talking about. So one of the groups in here is the Archaeplastida. And the Archaeplastida are different compared to a lot of the groups that we've talked about. The Archaeplastida are multicellular. They are marine. We wouldn't find Archaeplastida and red algae in Wolf Lake. Notice we talked about the brown algae, the kelp, a few days ago, and that's another group that's multicellular. If we look at red algae, the cells don't have centrioles. They don't have flagella at any part of the life cycle. They're also unusual because they have two layers of cell wall. They have an outer layer of something called agarose, and they have an inner layer of cellulose. Now, you might remember from Biology 105, plants have a cell wall, and one of the main chemicals in the cell wall of plants is cellulose. But plants don't have agarose. If you've taken a class in microbiology, that might look familiar with you. In microbiology, we use something called nutrient agar to grow bacteria on. If you've had a class where you've used Petri plates, Petri dishes, that's nutrient agar made from agarose that's in those Petri dishes. It's also a good example of how science has changed over the years. Agar first started being used to grow bacteria on during the 1800s. And when agar was discovered, or when it was used, I guess I should say, microbiologists were looking for something that would melt at a pretty low temperature. So something that we could melt and not necessarily kill the cells on it. And agar melts at about a little over 90 degrees. So it's not extremely hot compared to some things. But the microbiologist who was given credit for discovering it didn't discover it. It's something we often see in history. The person who actually discovered agar, or really who suggested it, she didn't discover it either, but the person who came up with this as being something that microbiologists might find useful was this guy's wife, because at the time, agar was used in canning. And if you go back to the 1800s, everybody canned their own food. So this woman suggested that her husband try this, and it worked. So he's given the credit for discovering it, but really his wife discovered it. And all you have to do to take it out of red algae is basically boil them. And the agar dissolves, because remember what I said, it has a low boiling point, around 90 degrees. And we can take it and purify it and use it to grow bacteria. Now, one of the characteristics of red algae is red algae are found inside lots of other cells of different groups of protistans. So remember, we talked about primary endosymbiosis, where a eukaryotic cell engulfs a photosynthetic bacteria. Secondary endosymbiosis, we mentioned, that's when another eukaryotic cell engulfs a eukaryotic cell that already has this bacteria incorporated into it as a chloroplast. Now, when we look at the red algae, a lot of times they're described as looking kind of like small evergreen bushes or small pieces of cabbage, but they are multicellular. Now, there are two groups of protista that are very closely related, called chlorophytes and charophytes. And they're grouped together as what are called green algae. And like most of the algae, they get their name because of their color. But let's go back to our first diagram. Here are the chlorophytes and the charophytes. Now notice, here's our red algae over here. So the red algae branched off from the green algae, and our fourth group in this large superclade is land plants. Red algae branched off first, so they're least closely related. Now notice that green algae is not a very good taxonomic group in terms of cladistics. Why? Because let's go back. Here's a common ancestor down here, and this is the common ancestor of the chlorophytes and the charophytes and the land plants. So when we take green algae, we're taking something from one branch of the tree and something from the other branch of the tree, family tree, and we're putting them in the same group. And according to cladistics, if we do this, then the green algae should include everybody in these two branches of the family tree. So it should include the chlorophytes, the charophytes, and the land plants. But we put land plants in a different group. But notice when we have something like this, when we have a group like green algae that doesn't include all the descendants of a common ancestor. So here's our common ancestor back here. All the descendants are the land plants, charophytes, and chlorophytes. If we have a group that doesn't include all the common descendants, this group is called polyphyletic. They evolved from two different common ancestors. So they evolved from the common ancestor here and the common ancestor up here of the charophytes and the land plants. So even though most biologists use cladistics to classify organisms nowadays, there are some of the rules that are broken pretty regularly. And one of the rules that's broken pretty regularly is demonstrated by green algae, paraphyletic groups, where there's more than one common ancestor, or where we don't include all the descendants of a common ancestor. So from the point of view of cladistics, that's not a good group. Now notice both of these groups do have a lot in common. They both contain chlorophyll A and chlorophyll B. And these are the same types of chlorophyll that are found in plants. Everything that's been discovered so far that's photosynthetic contains chlorophyll A. But lots of organisms don't contain chlorophyll B. They have a different type of chlorophyll, or they have separate chemicals altogether that aren't chlorophylls at all. So there are a lot of biologists, going back to our discussion of cladistics, that think that these two groups of green algae should just be classified as plants. That's what really makes the most sense. Now, if we look at these green algae, it turns out that green algae have a lot of different body shapes. Oops, Spirogyra is not on the next page anymore. So if we look at their body shapes, some of them are filamentous, which means they have long, stringy body shapes. So this is a type of algae called Spirogyra. And if you look inside the cells, so you can see the cell walls here. So the cells are basically rectangular. If you look inside the cell walls, the chloroplasts are arranged in strings that kind of twist around each other. So that's why it gets the name Spirogyra. As far as I know, this is the only type of algae that has a jazz band named after it. There's a jazz band called Spirogyra. Some of the green algae basically look like lettuce, or they're sometimes described as looking like leaves. For instance, an example of this is something called ulva. And actually, people do eat ulva, just like you could eat lettuce. Some green algae are just single-celled flagellated cells. But sometimes these single-celled flagellated cells group together and form colonies. A good example of this is something called Volvox. If we look at Volvox, all these little green dots are called vegetative cells. there's a kind of a matrix here, a kind of it's got some protein in it, some carbohydrate, and it forms a kind of a gum that all these cells are stuck into. And all these little cells, remember, these are colonial flagellated cells. Their flagella stick outward, and their flagella can beat. So Volvox moves, and the flagella aren't very well coordinated. So if we would be looking at Volvox under the microscope, Volvox would kind of roll like a ball. It doesn't move very efficiently. Now you can see a couple of clumps of big darker green blobs, and these darker green blobs are reproductive cells, and they can divide by mitosis, and they can form a new colony that breaks free from this matrix that's holding all the cells together. Volvox can also have cells that specialize and produce eggs and sperm. And then it can release these gametes into the water, and they fertilize one another. You know, if we go back to our original drawing, notice that the charophytes branch off from the land plants later than the chlorophytes. So the charophytes are more closely related. So we want to keep that in mind. And we're just about ready to start our last big group, our last superclade of protists. But I think we'll wait until tomorrow to do that. Are there any questions before we leave this set of notes? No?
Professor?
Yes.
I was wondering when the paper is going to be due.
What's today? Wednesday? Probably next Thursday is when it will be due.
Okay, thank you.
So I'll have something posted either today or tomorrow, because that's when the other thing that will be due the same day is the bacteria questions. Well, bacteria and virus questions. That's all one section. Any other questions?
If I can get a draft set, if I can like get a draft before it's due, can I send it to you and see if it's like
Yeah, that's no problem. I will give you some suggestions. I would say the only thing is, you know, make sure you get it to me by next Monday at the latest, just because depending on what else I have to do, it may take 2 days to get to it. So if you send it to me Tuesday or Wednesday, I just may not get it back to you in time.
Okay, thank you.
You're welcome.
Did you see my email by any chance?
No, I haven't seen it yet.
Okay.
I'll check it after, as soon as we get done, unless you want to talk about it now.
All right.
Are there any other questions or comments?
Quick question,
Sure.
Dr. Griffith. So we just keep working on our paper, and then when you load in the bacterial questions, we just start working on those?
Yeah, and for the final version of your paper, you can either send it to me separately as a word processing document, or if you want to, you can just cut and paste and put it in. That'll be the short answer question for that set of questions.
Okay. Thank you so much.
You're welcome. Any other questions or comments?